and it's a great opportunity to be able to present our work here at this really exciting forum. So can I just confirm if you hear me well and my slides are on the screen?
Yes, we hear you.
Thank you.
Yep.
Thank you. So the title of my talk today is Metabolic Reprogramming by Mesenchymal Stromal Cells as a Strategy for Lung Repair. And I would like to start by giving you a brief introduction to note this seminal publication by Spies and colleagues published in PNAS in 2006, which was the first report demonstrating that mitochondrial transfer from mesenchymal stromal cells to the mitochondria depleted A509 cells was able to rescue their aerobic respiration. Mitochondrial dysfunction is driving pathogenesis of so many conditions, and especially in case of lung conditions. And since this report, there was a strong interest in exploring this approach to attempt to deliver exogenous healthy mitochondria to the injured cells. And to date, from the data of multiple groups and many investigators across the world, we have very solid evidence that mesenchymal stromal cells indeed are capable of active transfer of their mitochondria to the multiple cells in different tissues. They are capable of doing that through two mechanisms which are being identified to date. So this would happen either through establishment of the direct cell contacts through establishment of the cytoplasmic bridges between MSC and the recipient cells, and then mitochondria would be transported along the actin microfilaments with the help of the complex protein machinery of motor proteins, of which MIRO1 probably is the most well studied. Another mean of mitochondria transfer is the ability of MSC to package their mitochondria into the extracellular vesicles and deliver mitochondria that way. In both scenarios, mitochondria uptake and internalization by recipient cells results in improvement of their mitochondrial fitness, enhancement of the levels of oxidative phosphorylation, improvement in ATP production, and restoration of function. So my research interest for the past more than 10 years was focused around development of mesenchymal stromal cell-based therapy for the treatment of acute respiratory distress syndrome, or ARDS. And that is a clinical disorder characterized by diffuse inflammation in the lung, leading to impaired gas exchange with systemic release of inflammatory mediators, causing inflammation, hypoxemia, and frequently multiple organ failure. It is the leading cause of death and disability in the critically ill, with mortality up to 40%. It could arise from multiple causes, but pneumonia and sepsis are the most common and the most devastating. It is characterized by multimodal pathophysiology. It is very heterogeneous disease, but the common features are dysregulated inflammation in the alveoli spaces, accompanied with disruption of epithelial and endothelial barrier, leading to flooding of the alveoli with the protein-rich edema fluid, and also infiltration of the inflammatory cells, which would eventually lead to impairment of the gas exchange. And this could be very often complicated by infection. Despite decades of research, there is no specific pharmacological treatment. And importance of this condition to public health has been strongly highlighted during ongoing COVID-19 pandemic, because COVID-19 patients have a high risk of developing this condition. And because there is no specific treatment, those who develop it are at high risk of death. So interest in investigation of therapeutic potential of mesenchymal stromal cells for treatment of this condition has been strong, and the studies started about 12 years ago. At the moment, studies by multiple investigators from multiple groups in different preclinical models have assembled a solid body of evidence that MSCs indeed have strong therapeutic effect in the preclinical models of RDS. And their effects target all the aspects of RDS pathophysiology. So they improve survival, they home directly to the lung, they have strong anti-inflammatory properties, they're able to restore endothelial and epithelial barrier integrity, enhance alveolar edema fluid clearance, and have antimicrobial properties. At the present, they already entered the stage of the clinical development and are being tested in the early stage clinical trials, including for COVID-19 induced RDS. However, to effectively translate MSCs into clinical application, we need better understanding of their mechanism of action. And although that majority of mechanistic studies to date were focused on the investigation of their ability to secrete multiple paracrine factors, it is clear that this secretion is just a fragment of much more complex system of interactions between MSCs and the host cells, and their ability to contact with the host cells through direct cell contact and organelle transfer, as well as secretion of extracellular vesicles, are being more and more increasingly recognized. And that investigation of the therapeutic mechanisms of MSCs in RDS is the focus and the central focus of the research which we are doing in my laboratory. So in this presentation, I'm going to talk and to present to you the results of our investigations on the role of mitochondrial transfer in MSC modulation of macrophages. And that was done as a part of the work where we investigated the mechanisms of anti-inflammatory effect of MSCs. And I will finish with the more recent data, which are still unpublished, where we investigated the role of mitochondrial transfer in MSC modulation of alveolar capillary barrier in RDS. So the first project which I wanted to present to you today was focused on investigation of MSC modulation of macrophages, and we wanted to investigate how they modulate them through direct cell contact. And that's how we discovered that they communicate with macrophages using tunneling nanotubes. So for the in the interest of time, I will present only the key data because all this is published. So this is the image of direct co-culture of primary human monocyte-derived macrophages stained in blue with human bone marrow-derived MSCs. And they were pre-stained for their mitochondria using mitochondria-specific fluorescent dye, mitotracker red. As you can see, MSCs form these long, thin structures, protrusions, which can reach in length up to 200 micrometers. And they are highly positive for mitochondria. So these are structures which are termed tunneling nanotubes or TNTs, and they have been described previously. And they serve as channels or tunnels, if you will, and for mitochondrial transfer. And mitochondria are transported along the structures to the recipient cells, in this case macrophages, and being readily taken up and internalized by macrophages. So that was really exciting finding. However, the next question was, so what is the functional outcome of this? And because the primary biologic function of macrophages is phagocytosis of microorganisms, and this is a highly energy consuming process, we thought that perhaps mitochondrial transfer will facilitate macrophage phagocytic activity. And indeed, when we gave mitochondrial labeled MSCs into the mouse model in vivo, we found that alveolar macrophages, which were positive for human MSC mitochondria, indeed demonstrated higher phagocytic activity. So that partially confirmed our hypothesis But the next stage was to test what role in this effect has transfer of mitochondria through tunneling nanotubes. And to approach this question, we blocked tunneling, formation of tunneling nanotubes in MSCs by pre-incubation in the small concentrations of the cytokalasin B. And that resulted in both abrogation of the effect of MSCs on facilitation of macrophage phagocytosis in the primary human macrophages in vitro, and also in MSC ability to improve bacterial clearance in the E. coli-induced pneumonia model in vivo. So the next project, which was taking place also almost simultaneously, was looking at the mechanisms of MSC modulation of macrophages through cell contact independent mechanisms, through paracrine mechanisms. And in this project, MSCs and macrophages were, again, primary human macrophages, were co-cultured in the transworld system without, which would preclude cell contact. And to most closely mimic RDS environment, they were stimulated with the samples from the bronchoalveolar lavage fluid of IRDS patients. So in that model, MSCs demonstrated strong ability to reprogram macrophages towards a more anti-inflammatory M2-like phenotype, as reflected in their suppression of secretion of pro-inflammatory cytokines, upregulation of the M2 phenotype markers, and it also was associated with the higher phagocytic activity, which is consistent with the data which we have seen previously. So to investigate the role of mitochondrial transfer through extracellular vesicles in that, we first needed to test whether or not we can detect any vesicles in MSC conditioned medium. And at that time, that was about 4 years ago, we just applied conventional flow cytometry. But even by this really crude method, we were able to detect that MSC conditioned medium indeed contains good number of nano-sized particles. And what was even more interesting, that about 25% of those nano-sized particles were mitochondria positive. We also were able to visualize presence of the mitochondria in MSC conditioned medium. So for that, we pre-stained MSC with MitoTracker Red again and collected this medium, and we were able to visualize mitochondria in this medium. We also were able to visualize mitochondrial transfer through medium to macrophages, as you can see here. So to proceed further, sorry, with functional experiments, we needed to find a way, a good specific way, how we can inhibit secretion of these mitochondria containing vesicles specifically. And we found that pre-incubation of MSC with rhodamine 6G would produce us the desirable results. So this treatment resulted in inhibition of MSC mitochondrial respiration and ATP, mitochondrial ATP production. However, that was compensated by glycolysis. Importantly, that was not toxic to the cells, but what is even more important for us, that this treatment did not significantly affect ability of MSCs to secrete other paracrine mediators, which were demonstrated to be important for their functional effects previously. So we were confident that that approach would provide us with the conditioned medium, which would specifically be depleted from mitochondria. So the next question was whether or not there is any effect on mitochondrial respiration and macrophages. And this was assessed by measurement of oxygen consumption rate by Seahorse metabolic analyzer. And MSC conditioned medium was capable to upregulate both mitochondrial respiration and mitochondrial ATP production by macrophages. However, this was this was not the case in when macrophages were incubated with conditioned medium from rhodamine pretreated MSCs, again confirming that this conditioned medium do not contain functional mitochondria. And finally, conditioned medium which was depleted from mitochondria was not able to recapitulate effect on macrophage polarization, suggesting that it is mitochondrial transfer in extracellular vesicles, which is an important mechanism responsible for MSC macrophage polarization in the RDS microenvironment. So all this work was done to investigate the mechanisms of MSC immunomodulatory effect. However, another important pathway involved in the RDS pathogenesis is disruption of alveolar alveolar capillary barrier. And that was the focus of the most recent project, which I'm going to present now. And these data are still unpublished. So we started to this project with the better characterization of our EV preparation. And by nano-tracker analysis, we were able to demonstrate that our EV preparation predominantly consists of the small size particles, about 100 to 300 nanometers, which would fall into the category of exosomes. However, interestingly, it also contains substantial fraction of vesicles with the size range of 500 nanometers and more, which would fall into the category of where mitochondria would be located. This data was also confirmed by electron microscopy, by which we were able to observe presence of small exosomes, larger multivesicular bodies, and structures of 501 micrometers in size, which resemble morphologically mitochondria. Mitochondria presence further was confirmed by expression of the mitochondria-specific protein TOM20, which is a mitochondria outer-membrane transporter, and EV preparation has a relatively high expression of this protein. Also, by bead-assisted flow cytometry at this time, we were able to detect that at least about 20% of our population of EVs are double positive for mitochondria and for surface markers of the MSC plasma membrane. We also conducted characterization of the EVs generated from rhodamine-pretreated MSCs. And interestingly, we were able to see that they, although they contain this fraction of the small size particles of the exosomes, the vesicles with 500 nanometers and higher, which would correspond to mitochondria, were absent. And this was also confirmed by Western blot, where rhodamine EVs did not express mitochondria proteins. So that again made us confident that rhodamine-pretreated MSCs do not secrete mitochondria, and we used these rhodamine EVs in consequent experiments as a control for the presence and effect of the mitochondrial function in our EV preparation. So because we were interested in the effects on the barrier functions, we needed to test whether or not endothelial and epithelial pulmonary cells will be able to take up MSC-derived mitochondria. And in order to do that, again, we pre-stained MSCs with MitoTracker red and then isolated EVs from this pre-stained MSC population, and then applied these EVs onto the primary human endothelial and primary human pulmonary microvascular endothelial cells, or primary human small airway epithelial cells. So both these cells play a role in RDS pathophysiology. And as you can appreciate from these images and as well as the movie, there is a robust uptake and internalization of MSC mitochondria by both cell types. So the next step was to investigate the effect on the mitochondrial respiration in these primary pulmonary cells. And again, this was done using Seahorse metabolic analyzer. And as we observed that inflammatory stimulation by LPS resulted in significant inhibition of mitochondrial respiration and ATP production, which was restored by MSC EVs. However, that was not, EVs without presence of mitochondria were not able to do so, again confirming that this is a good control for us. And then we assessed the barrier functions, and that was done using Excelogen technology, which measures cells' electrical impedance. And as expected, LPS induced pronounced barrier disruption in both cell types, which was restored by MSC-EVs, and that effect was dependent on, critically dependent on the presence of mitochondria in the EV preparation. So that was all good data. However, this was done in the simple monocultures, and we wanted to investigate whether or not this will be still relevant in more clinically relevant model of whole human lung tissue. And in our lab, we have a privilege to get access to the human lungs rejected for transplantation. And to maximize the use of these lungs, we have established this model of ex vivo culture of precision lung slices. And the beauty of this model is that you can keep these slices in culture for weeks, and they preserve all the native architecture of the human lungs, and you can do multiple interesting things with them. So we first wanted to confirm that inflammatory stimulation would induce mitochondrial dysfunction at the level of the human lung tissue. And we assessed this by measurement of the mitochondrial membrane potential using JC1 fluorescent dye here, and mitochondrial ROS production using specific MitoSOX fluorescent dye. And both parameters were dysregulated with LPS stimulation, and indicating significant mitochondrial dysfunction. However, this was restored with MSC vesicles and not vesicles generated from MSC with dysfunctional mitochondria, suggesting that it is mitochondrial transfer which is responsible for restoration of mitochondrial fitness. And also this data was consistent with the data on mitochondrial respiration in these slices, where LPS resulted in significant inhibition of mitochondrial respiration and ATP production, and that was restored by MSC-EVs, and presence of mitochondria was critically important for that. And finally, we wanted to test whether or not this will be also true for the whole organism level, and we applied in vivo mouse model of LPS-induced lung injury. So in this model, MSC-EVs, and we used 2 different doses of EVs, demonstrated strong therapeutic effects, but most importantly, they were able to significantly decrease a concentration of the bronchoalveolar lavage protein, which is an indicator of alveolar capillary permeability. And also there was a pronounced anti-inflammatory effect with MSC-EVs. Importantly, their effect was again critically dependent on the presence of mitochondria, as rhodamine EVs were not effective. We also wanted to test if this is related to the impairment of mitochondrial function in the lung tissue. And to test this, we again applied precision cut lung slices approach. So in this model, after mice were injured with LPS, then treated with EVs, we collected, next day we collected their lungs and prepared precision cut lung slices, and then measured mitochondrial respiration in those slices generated from the in vivo model. And the data in this in vivo model are very consistent with the data which we see in human lung tissue slices, where LPS results in significant inhibition of mitochondrial respiration and ATP production, and that is restored by EVs and critically depend on mitochondrial transfer. So to conclude, MSC transfer functionally active mitochondria to recipient cells through nanotubules and extracellular vesicles. Mitochondrial transfer enhances recipient cell oxidative phosphorylation, and in inflammatory environment, mitochondrial transfer leads to metabolic reprogramming of macrophages towards anti-inflammatory phenotype with enhanced phagocytic activity and improvement of epithelial and endothelial barrier integrity. So I'm finished here. Thank you so much for your attention, and I would like to acknowledge my group, my founders and my collaborators. Thank you so much again, and I'm happy to take questions.